We are going to take a look at meiosis. This is the type of cell division that produces gametes or sex cells. Unlike mitosis, that produces cells that are identical to the original cell, meiosis produces cells that are have half the original amount of DNA. So mitosis starts out with a 1,2N cell and produces two 2N cells. And meiosis starts out with one 2N cell and it produces four 1N cells. And these 1N cells can become either a sperm or an egg, depending on the gender of the parent that they occur in. They occur in the gonads or sex organs of the adult. So in human males, they would occur in the testes, and in human females, they would occur in the ovaries. And also what's interesting is that these 1N cells are not identical. We should make a note of that. Not identical to the original cell. They're different. And this is good because it increases variation. And we are going to, as we draw it, we're going to need to use color to draw meiosis. So we are going to start out with prophase 1. And again, this is prep phase, just like in mitosis. So we can draw our cell. And we can draw the centrioles starting to form. And in the animal cell, these will help to guide the chromosomes. Spindles might start to form. These are the muscles of the cell, little microtubules that can grab onto the chromosomes and move them around. And our nucleus will start to dissolve. And then inside the cell, excuse me, we are going to draw 3 sets of chromosomes. And we can use red for the chromosome from the dad. And we can use, we'll draw 3 different sizes. So there's our red chromosomes. And then we'll use green for the chromosome from the mom. So notice that we have 2 versions of chromosome 1. So the red one and the green one that are both large, 2 versions of chromosome 2, which are the medium size, and 2 versions of chromosome 3, which are the large size chromosomes. So next up is metaphase 1, and this is the really exciting phase. This is when a lot of good stuff happens. Okay, so in metaphase 1, we start out with our cell. We've got our centrioles. And the spindles have now moved our chromosomes to the center of the cell. And we have chromosome 1 right here, chromosome 2, and chromosome 3. These should be connected like the X shape. And the homologous chromosomes are attached And so we wind up with this structure, and the structure is called a tetrad. So I think we'll use a highlighter, and we'll just highlight to make our comment on that. So the tetrad is 4 homologous chromatids, or 2 homologous chromosomes. Connected. And this has a name in Metaphase 1. It's called synapsis. So synapsis is when the tetrads form. So something interesting can happen here during synapsis, and we'll demonstrate this on chromosome 1, and that is that the chromosomes, when they're really close together like this, they can actually swap segments of DNA. So the end of this red chromosome on chromosome 1 can actually swap over to the green, and some of the green can swap over to the red. So remember, you get 1 chromosome from your mom and 1 chromosome from your dad. So here, those 2 chromosomes swap. So your mom's chromosome now has some of your dad's genes on it, and your dad's chromosome now has some of your mom's genes on it. And this actually is neat because it increases variation. So we'll call this right here crossing over. And it's when homologous chromatids swap DNA. And this will only happen during synapsis because it's the only time that they're close enough to do that. And this is good because it increases variation. Instead of having 1 chromatid just always be the same, now it has some genes from a homologous chromatid on it. So this increases variation. So now we are going to go on to anaphase 1, and I think we'll continue on the next page. And I think what I'll do is I'll actually continue this in the second video. So I'm going to stop this now, and I'll pick up with you on second video on meiosis.